For centuries, humans have attempted to create immortality stones and potions, but what if the key to increasing the human lifespan lay in our non-coding DNA?
Wait, what?
In humans, telomeres, located on linear chromosome ends, are genetic aglets made up of thousands of the base sequence TTAGGG bonded to the protein complex shelterin and act a little like the protective tips on shoelaces. Telomeres help cell division run smoothly. Without telomeres, DNA repair mechanisms would see chromosome ends as double-stranded breaks and stick all chromosomes together, triggering apoptosis. So, how are telomeres associated with aging? Well, it's all to do with the end replication problem. DNA polymerase III needs an RNA primer and moves in the direction 5' to 3'. DNA is antiparallel, so the leading strand is formed in one piece while the lagging strand is created in short Okazaki fragments. When the lagging and leading strands are complete, polymerase I replaces the RNA primers with DNA nucleotides, except for the RNA primer at the end of the lagging strand because it can't move from 3' to 5'. This results in a single-stranded overhang. Therefore, every time DNA is replicated, telomere length becomes shorter. Eventually, telomeres reach a critical length in which there is not enough telomere to bond with the shelterin. Replicative senescence is triggered by the critical length to prevent cell malfunction. Having too many of these senescent cells increases the rate of aging, as tissues with inactive cells cannot easily repair themselves. Telomerase ensures that telomeres don't reach senescence too quickly by adding TTAGGG sequences onto the 3' ends of telomeres after cell division. Telomerase is not active in most somatic cells, but has high activity in other cell types. Gametes have high telomerase activity to ensure that offspring have sufficiently long telomeres. Hematopoietic stem cells have high telomerase activity because they differentiate into blood cells. However, after senescence, they differentiate into fewer blood cells, including white blood cells, making elderly people more susceptible to disease than younger people. Cells may become cancerous when telomere length reaches 2 or below the critical length. This triggers a rapid increase in the activity of telomerase, which contributes to the uncontrolled proliferation of cancer cells. External factors like smoking and chronic stress also cause telomere shortening, and therefore quick aging. It is evident that telomere shortening acts as a genetic clock that limits the lifespan of humans, but if we manage to stop this clock, we would be one step closer to immortality.